All right. Well, good morning, everyone. Good afternoon, good evening, and welcome to the webinar on scientific information in the Biosafety Clearinghouse. My name is Zakir Jaffrey, and I'm the Program Management Officer of the BCH. And I'm joined today by my colleague, Austin McLaughlin, who is the Associate Program Management Officer, and he upkeeps a lot of the scientific information in the clearinghouse. So you'll be hearing quite a bit from him. So as many of you may be aware, we recently conducted a survey on training needs on the new platform. And the number one request we received from respondents was for the Secretariat to host a webinar on the topic of registries as well as scientific information in the clearinghouse. So in response to that request, would like to spend this next hour with you all today, walking you through what kinds of scientific information exists on the platform, how you can find it, and how you can submit or register this information to the BCH. So as part of the registration process, we also took questions from participants, which we will field at the end of the presentation today. So if you have any questions as we go, please feel free to enter them into the appropriate questions box, which I believe should be at the bottom right of your screen. So please feel free to do so as we go, and we'll take those questions at the end. Also note that this webinar is being recorded and it will be shared on the YouTube channel of the Secretariat as well as through the BCH. So in the knowledge base of the BCH, we have an article where we share all our webinars, and this will be added as well. So you'll be able to access it for your future reference. So with that, we can go ahead and get started. All right. So before we dive into the topic of the scientific information in the platform, we just want to take a step back and just point out some of the context of the BCH. So I'm sure, as many of you know, the Biosafety Clearinghouse was established by Article 20 of the Cartagena Protocol as the central mechanism to facilitate the exchange of scientific, technical, and legal information on living modified organisms, or LMOs. So the BCH is this instrument that allows parties to share information on their use and handling of LMOs, and really, therefore, plays a fundamental role in facilitating the implementation of the Cartagena Protocol. So I mentioned Article 20. Let's just take a look at some of the key or all of the clauses within that. So Article 20 is on information sharing and the Biosafety Clearinghouse. And as you see right at the top, what I mentioned in the last slide for Article 20, Clause 1A is to establish the BCH to facilitate that exchange of information I just discussed. And you see written there. So that's a key element. We also see in 1B, it's to assist parties to implement the protocol. So parties have very specific obligations that are set out throughout the protocol, and the BCH really has to function in a manner that allows parties to meet those obligations. And that really includes the need for the BCH to host comprehensive scientific information, which we'll talk a little bit more about in a moment. So Clause 2 is to provide access to information that's made available by parties. So it's really about making the above information available for other parties, but also the greater public through the BCH, and to connect with other biosafety databases. So the key highlight that I mentioned in the last slide, and this one here for our conversation today, is really the need for the BCH to facilitate this exchange of scientific and technical information. So let's look at the last couple clauses for Article 20. We see for 3, it really outlines some of the key requirements that inform some of our necessary records of information on the platform. So Austin, my colleague, will talk about this in a moment, but on the BCH, we have 2 main categories of information. We have national records, which are managed by parties, and we have reference records that are managed by us at the Secretariat. So essentially, a lot of those national records are requirements that are set here. We see in Article 20, but also throughout the protocol. So we see each party shall make available to the BCH any information required under the protocol, including any existing laws, regulations, and guidelines for implementation. So we have a law format for that. any bilateral, regional, and multilateral agreements, summaries of its risk assessments or environmental reviews of LMOs generated by its regulatory process. So we have a risk assessment national record format as well as a reference record format, and its final decisions regarding the importation or release of LMOs. So we have a decision format for that as well. So if parties are required to make information on their laws, decisions, and risk assessments on LMOs available, then the BCH really needs to support that requirement by containing comprehensive and up-to-date scientific information on these LMOs. So today we'll really focus specifically on this scientific information and provide details on the kinds of information the BCH provides and how you can find it. Then I'll quickly also mention Article 20, the fourth paragraph or clauses on the modalities of operation, which really just highlights how the BCH needs to function to meet those needs. So with this now, I'll pass the floor to my colleague, Austin McLaughlin, who I mentioned earlier. He's really the key officer in charge of publishing, updating, and validating the scientific information in the BCH. So Austin, feel free now. I'm going to stop sharing my screen and pass the floor to Austin, and he'll share the screen for his presentation.
Thank you very much, Zachar. And as I get going here, I also just wanted to say good time of day to all of you who are joining us online, and thank you so much for having an interest in the BCH and the scientific records. So before I begin, can you see the presentation?
Yeah, we can see it well.
Perfect. So with that, I'm going to begin. So today I will be talking about the scientific information on the BCH, and I'll show you how to search for that scientific information, as well as show you what some of the information looks like. But before I begin, and sort of building off what Zachar just said, there are two types of records or information types on the BCH. So there are national records, which are managed by the governments, which include information parties are obligated to provide in accordance with the protocol, and as well as other national information that might be relevant to the implementation of the protocol. Then there are reference records, which include biosafety-related resources and information. This type of information can be submitted by any user, and they're validated by the Secretariat prior to the publication. So with that in mind, and when considering what scientific information is on the BCH, so for the purposes of this presentation, when we refer to scientific information, we're referring to mainly living modified organism records, genetic elements, organisms, biosafety virtual library resource, laboratories for detection and identification of LMOs, risk assessments generated by independent or non-regulatory processes, as well as risk assessments generated by regulatory processes, and biosafety experts. So in terms of the information in the BCH, we are proud to say that we have over 17,000 records on the BCH, including almost 1,000 LMO records, over 840 genetic elements, over 260 organism records, over 70 laboratory records, over 1,500 virtual library resource records, over 2,600 risk assessments, and over 360 biosafety experts. So this is just to highlight that the BCH is a comprehensive database of biosafety-related information. So with that in mind, I thought it would be good to sort of go through how to find some of the information on the BCH. So of course, you would start by going to our website, and then you'll see at the top of the screen, you have a few different options. So you have a home, about, search, submit, country profiles, help, and forms. So if you were to click on the search, you'll have this drop down that shows up. And if you click on the records, you're able to then see the next set of filters for the records. So it'll look like something like this. So in terms of the record types, it's pre-divided into national records and reference records. And I just want to highlight here, although you can see that there's information on national focal points and biosafety laws, because we're talking about scientific information, I just wanted to highlight that right here. So you're able to filter by clicking the black box and then filtering by the type of record that you're interested in. But before I continue, I also want to highlight some of the other global filters. So to start with, we have keywords. So across the different record types, there are various types of boxes that are ticked. So perhaps the keywords might be useful for you if you're searching for information related to, in this case, agrobacterium mediated DNA transfer or algae. So across the different records, there's keywords that are pulled out that may facilitate the search. Next, we have countries. So if you're interested in information related to particular countries, this is more for mostly for the national records, but you're able to search by countries. And just to highlight here that there are both information from both parties as well as non-parties that is made available on the BCH. You can also search by region. So if you're interested in, for example, a particular region, you're able to find information on that. Finally, we have our date filter. So this is really important to highlight because this is the date that the record was published on the BCH. For some record types, such as risk assessments or virtual library resource records, there's a date associated with when the risk assessment was done, or perhaps when the scientific publication was published in a peer-reviewed journal. That is not the date filter here. I would really like to stress that I'll get back to this later, but this date filter in the global filters is solely for when the records have been published on the BCH. So to show you sort of how to search for information, I thought it would be good perhaps to search for a living modified organism. So if you were to click that, your search will be refined and you'll get a result like this. So when you first get your results, the default is to have it sorted by the last updated. So the list that you see here would be the most recently updated or published LMO records. So then looking at the top, you have your search bar. So if you are someone that likes to search similar to Google or to another search engine like Bing, you can use the search bar here and you're able to enter in your keywords or your search to filter through the records. But there are also sub-filters. So the sub-filters are really powerful because they are specific to the type of record themselves. So in terms of the LMO record, they're related to the common format that is used for the LMO records themselves. So you can see here that you have a free text box, so you're able to search for keywords similar to the global one, but this will search all of the record in the LMO record themselves, all the text. Then you have your common uses, so whether this is for food or feed, your techniques used for the modification, so for example, agrobacterium-mediated transformation. You have your modified traits, so if you're interested in, for example, herbicide tolerance or insect resistance, followed by the unique identifier, so this is the OECD unique identifier, then the genetic elements that are present in the LMO. So if you're interested in the cauliflower mosaic virus promoter, and then you have the recipient organism, both by common name and scientific name, and the donor organism also by common name and scientific name. I also just want to highlight that compared to the previous BCH platform, there is improved functionality, so you can really refine and filter your results in a more powerful way just by clicking on the gear symbol up there. But just to show you how the subfilters work, we will say we're interested in a modified trait. So when you click on that, you will have this pop-up that appears, which gives you all of the possible filters for the possible modified traits that an LMO may have within the BCH. So for example, if we were interested in resistance to Lepidoptera pests, we will click that and you will see that the parent and the children, so in this case, the parent being Lepidoptera and the children being cotton bollworm, European corn borer, and fall armyworm are automatically selected. So if you were to click apply, your result now is filtered by LMOs that have the corresponding modified trait that you're interested in. So all of these ones here are have the resistance to Lepidoptera pests. So just as I mentioned before, and just to highlight that these are sorted by the last updated, but you're able to use the box up there that I've highlighted on the screen if you want to change the way it's displayed or to sort in a different manner. But you're also able to share the search results. You can also export the search results. And I also wanted to highlight that you can save the search results. So if you were to click on that, you will get a pop-up. So you're able to enter the title of your search, as well as you can actually select an option to email you when new information is added relevant to your search results. So if there are new LMO records that are added that have, for example, Lepidoptera insect resistance, you will receive an automated email saying that there has been a new LMO added to the BCH. Otherwise, your search will be saved under this My Save Searches function. So you're able to go back to your search, and when you click on the search, it'll regenerate the search results for you. So now I think it would be maybe a good opportunity to go through what an LMO record looks like. So the type of scientific information you'd be actually perhaps looking for and viewing for. So if we were to scroll down, and we were on our search results and we selected this modified cotton, this is what our LMO records look like. So immediately you can see at the top, you have options to generate a PDF. You have an option to print the record. If there are other languages, if the record has been written in other languages, sorry, you're able to toggle between those languages. You can also share the record, and then you can compare to previous versions. So in comparison to the old BCH, in the old BCH, there were 3 tabs, one being the LMO information, then there is one for decisions, and there's one for risk assessments. This has been maintained on the BCH, but looks a little bit different. So at the top of the record, you'll see that there's links to the decision and the risk assessment. So if we were to say click on the decisions, a new tab will open up on your browser, and you can see all the countries that have taken a decision on this particular LMO. So we have BCH icons that you're able to move your cursor over and sort of see what it means to see how what type of decision was made on that particular LMO. But I also wanted to highlight that we have maintained the link to the CropLife International Biotrade Status Database, so you can also get information on that's published there associated with the LMO and the countries themselves. So just going back to the LMO record themselves and working our way down the record, the next thing you'll notice are our LMO quick links. So this is a label that's used to facilitate work of customs officials and can be used in documentation. So this has the OECD unique identifier and the name of the LMO, and then this QR code. So previously this was a barcode, which you would need a barcode reader to scan, but we've replaced this with a QR code. So since everybody's using smartphones these days, it's scannable by a smartphone and takes you directly to this particular page on the BCH to get information on this particular modified cotton. Then moving down the record, we have the name of the cotton, we have the transformation event, then we have the OECD unique identifier, and then information on the developer. And then moving down, we have a box that is called description. So in general, we think of this as like an executive summary of the LMO. So this includes the information on the organism itself, the traits that are expected, and if perhaps a protein that is also expressed. So just to sort of give you an idea, a general idea of how and what this LMO is. Following this, we have a link to an organism record, so cotton in this case, and then we have a field for point of collection. So generally what the Secretariat has been doing is we've been putting information on the particular cultivar, variety, or strain of organism here if it's available, just to fill out the records and to improve the completeness of the LMO records. Following this, we essentially have our molecular biology section. So this is the characteristics of the modification process. So we have information on the vector, so the name of the vector, how the LMO was produced, so the transformation process, followed by a genetic elements construct. So here I just wanted to highlight that the Secretariat has been working hard to build these into the LMO records to help facilitate the understanding of how the genetic elements relate to each other and how the LMO essentially works. So we include the base pair sizes here too, and it'll appear on the LMO record as these sort of genetic sets here. Following this, we have links to the introduced genetic elements, followed by a field for notes regarding the genetic elements present in the LMO. So in this box here, we include as much information as we can on how the genetic elements function, how they're related to each other again, as well as any other information that could be important for a regulator or a user of some sort. So this includes, for example, if there's information on the copy number or the insert, if there's been truncations or codon optimization, if there's specific expression in particular tissues but not others, that type of information we generally include here to sort of help you understand how the genetic elements are working. Moving down the record, the next part of the record are the LMO characteristics. So these are our modified traits. So it's good because we can see here that we have resistance to Lepidoptera pests, so it matches our search results, which is good. Then the common uses for the LMO, so in this case, food and feed or fiber and textiles. Then we have our detection methods section. So this is really important here to highlight that the BCH has automated links to the European Union Reference Laboratory for GM Food and Feed, so their detection methods, as well as the CropLife International Detection Methods database. So these are automated links that are generated based on the OECD unique identifier and are attached automatically to the record such that you have verified and reliable detection methods available to you. So then to sort of finish at the bottom of the record, we have an additional information section. So if there's other types of information that might be pertinent to this particular LMO or might be interesting, we'll include it here. And then the Secretariat works hard to ensure that we have reliable scientific information. So we include attachments to other databases, such as the Eugenius database, as well as if there are regulatory documents available, such that you have reliable scientific information. And this field also will work as essentially like a reference or a bibliographic reference section to the LMO record. So if you see information in the record and you would like maybe a little bit more information or would like to know the source of that information, we provide the references here for you to explore as well. Then we have at the bottom of the record the section that shows all the cross-linking between other records in the BCH. So any records that are referencing this record are displayed here. So you can see that if this is maybe involved in a stacked event, so we have other living modified organism records, we have risk assessment records, we have country decisions, we have laboratories that are able to perhaps detect this LMO, and then we have risk assessments also by independent processes. I also wanted to highlight that you are able to report. There's this report button. So if you find information that's maybe you're not sure of, or you think might need to be changed, or there's a typo, you're able to report the record to us and flag it for our attention. So just to sort of show you what the cross-referencing looks like, I will select, for example, the risk assessment. So this will expand out and you're able to see all of the countries that have taken risk assessment or have done risk assessments on this particular LMO. So you're able to sort by country or by the title of the record, the BCH identifier, or by last updated on. So just to maybe show you what a national record looks like and to show you what happens if you say click on a risk assessment record, we will use this example from the Philippines. So if you were to click on that, you'll be taken to our national risk assessment records. So the first thing you'll notice is that reference records are orange and national records are blue. But you will also notice that the country, since this is a national record, is displayed right at the top. So in this case, the Philippines. So then working our way down the record, you can see that there's the title of the risk assessment record, information on the competent national authority that performed the risk assessment, followed by the risk assessment details. So we have a link to the LMO record. So that's what's facilitating that cross-linking between the LMO record we were just on and this risk assessment record that we're on right now. Followed by this, we have the scope of the risk assessment. Then we have a link to the summary, the risk assessment summary or report. Following this, we can see that in this case, the Philippines has added additional information. So we have information on the methodology and the points to consider. So we have information on the potential adverse effects, likelihood that these effects might occur, possible consequences of these potential effects, recommendations on the risks, whether they're acceptable or manageable, and any management strategies, needs for further information, the receiving environment considered, and then information on detection and identification methods proposed. And then, of course, we have our additional information section, and then they have also included a link to the decision document as well. It's important to recognize here that although we have two risk assessment record types in the BCH, national and sort of the independent one or non-regulatory one, the fields are they're exactly identical, except that instead of a country being displayed, an organization will be attached to the record. So if you're looking for particular information for risk assessment records, it's important to understand how the subfilters work. So again, we have our free text box, so we can search all the text within the risk assessment records. We have a subfilter for the scope of the risk assessment records, so if it's for food or for feed. Then we have several selections that are related to the LMO being evaluated. So you could search by the LMO record itself, the modified traits, the genetic elements present, the techniques used for the modification, and then the parental organism, both the common name and the scientific name. It's also important to recognize here, and I told you I would get back to it, but just to highlight that we also have the date of the risk assessment record field right here. So here is where you're going to select if you are looking for a particular date range for the risk assessment. When the risk assessment was done, you use this date field. If you're looking for the one that was published on the BCH, you'll use the global filter section. But just to highlight the difference between the two date filters. So moving on to other types of records. So here we have genetic elements, organisms, and laboratories. So again, we have our free text box, which is useful for if you're searching for just using sort of similar to Google. And then for genetic elements, we have the category of the genetic elements. So if it's a terminator or a promoter or a protein coding sequence, whether it's a synthetic molecule, the traits that are conferred by the particular genetic element, the name of the genetic element, and then the donor organism, both the common name and the scientific name. For organisms, it's quite simple. It's the type of organism. So you can search by the plant, or if it's an animal or a bacteria, the domestication status of the organism if it's available, and then the common uses of the organism itself. For laboratories, we have a few more subfilters, but I just wanted to highlight here that there's a subfilter for services and activities performed. And this differs from the types of detection and identification methods available. So for the services and activities, these are things like sampling, validation, capacity building, where the types of detection and identification methods available are more your molecular techniques. So the DNA-based techniques, PCR sequencing, or the protein techniques, strip tests, ELISA. And then you have the other filters for if you're interested in the type of LMO, so whether the lab does analysis on plants or microbes, the geographical region of the laboratory, as well as the particular LMOs or genetic elements detectable by that laboratory. For virtual library resource records, since virtual library resource records could be, they're really all information that's related to biosafety. So this could be legal information, it could be public awareness information, as well as scientific information. And they do contain a lot of scientific information. You'll need to use the subfilters to find it. So you have a few options. One could be using the free text box. So for example, if you're interested, say risk management, you can use it to type in risk management. But you can also use some of the other filters, such as the biosafety thematic areas, or if you're looking for information on a particular LMO or organism or genetic element, you can also search for it using those fields as well. Also, I just want to highlight here that the publication date is the publication of the particular resource, so the database or the journal, for example, when it was published online or made available, not when it was made available on the BCH. That is related to the global filter. And I know I've said it before, but just to highlight that. So there's other types of subfilters you can use as well if you're interested in a particular language or a CBD subject area. There's also a field related to the previous guidance on risk assessment. So probably one of the more easier ways to find scientific information within the virtual library resource record would be to use the biosafety thematic areas. So if you were to click on that, a pop-up will appear. And if you scroll down, we do have a selection for scientific and technical issues. So the Secretariat does work to make sure that this is one of these selections or multiple of these selections, if related, are selected so that it facilitates that search to make it easier to find scientific and technical information within these types of records. So there are other ways to find information on the BCH that I would also like to highlight. So one of the other ways is the cross-referencing between records. I demonstrated this a little bit when we were looking at the risk assessment record. We started from an LMO record for the modified cotton, and then we were able to find risk assessments that were done on that LMO, either through the top where you had the link to the risk assessments or at the bottom where it shows all the cross-referencing. So this cross-referencing can be quite important. So if you're doing a search for a particular, say, genetic element, and you're interested in what LMOs may have that genetic element or laboratories, it'll be shown at the bottom of the record. So this cross-referencing and interlinkages within the BCH are really important and really powerful in finding scientific information on the BCH. However, we also maintain registries. So if you were to go at the top of the page under search, so instead of clicking records, you will notice that we have a full section for registries. So if you were to click on that, it's just to highlight that the Secretariat maintains 3 registries on the BCH. So these are compiled lists of all LMOs, genetic elements, and organisms found on the BCH. So just to sort of show you what these look like, here's the LMO registry, for example. So this is sorted first by OECD unique identifier in alphabetical order, followed by the scientific name of the organism, and then thirdly by the transformation event. So it's structured in a way that should facilitate finding information or scrolling through the list and easily sort of understanding how the information is sorted. We also have an export function that you're able to export the full registry to an Excel spreadsheet. For the organism registries, it's sorted very simply by the scientific name in alphabetical order. For the genetic element registry, it's sorted first by the name of the genetic element, followed by the scientific name of the donor organism. So actually, this is a good example right at the top. We have 3 genetic elements with the same name. So you might think this is a triplicate in the BCH, but however, if you look under the donor organism, you can see that there are 3 different donor organisms. So just before I wrap up, I just wanted to run through how to submit information to the BCH. So if you click on the submit at the top, it'll bring you to your dashboard. If you are a national focal point or a particular authorized user, you'll be able to see the national records as seen here. And just to highlight that if you want to submit a record online, you can use this plus sign that's within the green box on the left-hand side. But there's also the offline formats available in all UN languages, so you're able to fill in information there too. Otherwise, if you scroll down, or for users that don't have that, if you're a regular user, you'll just see the reference records. So here you're able to submit information on any of the reference records that we have and send it to the Secretariat for validation. So perhaps if you're sending information to the Secretariat, I'd like to maybe give you a few tips for best practices when submitting information. So the first one would be to fill out as many fields as possible. But just to note that the mandatory fields, which are marked with a red asterisk, will need to be completed prior to the submission of the record. The BCH will not let you submit the record unless you filled out all the fields. Another tip that I would like to provide is that it would be good to include the source of the information. It acts as a reference to support the information that you're submitting to us. So this could be links to publications or attachments. The Secretariat will also add and try to fill out the records themselves, but this would help us in our work. I'd also like to suggest that when providing sort of links to peer-reviewed journal articles or other publications, it would be really good to use the DOI link. This future-proofs a bit the BCH, because these links are, the idea behind them is that they won't be broken in the future. And as URLs change sometimes from time to time, or links sort of don't work anymore, this sort of works to maintain that link to the particular publication. For LMO records, if possible, a vector map or a table of genetic elements is always very much appreciated. And it just helps with the descriptions of the LMOs and creating those genetic element constructs within the LMO records themselves. So with that, I think I would like to say thank you so much for your attention. I know it's a lot of information that I've provided, but I'm looking forward to, if you have any questions, I'd be happy to answer them. But just thank you so much for your attention. And yeah, I'll hand the floor back to Zachary. Thank you.
Thank you very much, Austin, for that comprehensive run-through of the scientific information in the BCH. What I'll do now is just share on the screen some of the questions we received through the registration process for the webinar. And I will say again, if there were any questions that anyone had through the presentation, please feel free to use the Q&A box to add them here. But in the meantime, we'll go through some of the questions that have already been asked. And because you all had these questions in advance of the webinar, I think several of them have already been answered, or majority of them, I think, through Austin's presentation. We have organized them here. So the first batch that you see are questions related to publishing scientific information or editing. And then the second batch of questions you see closer to the bottom seem more related to how to best search and find information. So we see quite simply the very first question at the top is how to upload information on the BCH website and other related issues. So this question, I think, speaks directly to what Austin had shown just in his last few slides. I'll quickly show the BCH here. The key place to go to upload information is Submit. And as Austin well pointed out, if you're a national focal point or a national authorized user, here you'll be able to create and manage your national records. But all users will be able to also submit reference records that you see below there. So it's in this area where you can access the submission forms. And I do want to point out that at the end of February, we hosted a webinar on the whole process to publish information. So if you're unsure about that full process, we encourage you to view that webinar that is available on YouTube. And I might ask one of our colleagues to maybe share it in the chat. So if anyone wants to review that information, please feel free to do so. But Austin mentioned you press the plus button, and then that will take you to the submission form where you can follow the steps to enter the mandatory information and other details. So the second question is how to register LMOs that have not been issued with a unique OECD unique identifier. So I'll quickly, maybe I'll let Austin speak to this as I'm pulling up the submission form and showing exactly where that question is.
For sure. So you are able to register LMOs that haven't received a unique identifier. As we know, these OECD unique identifiers are generated for LMOs that have been commercialized. But there is a field here that Zachary is showing on the screen, and it is a mandatory field. You have an option to select yes or no. So if it doesn't have a unique identifier, you could select no. If you select yes, a new field will pop up and you're able to enter the unique identifier. If this is a commercialized event and you think it should have a unique identifier, one option would be to actually talk to the developer to see if they've developed a unique identifier. And OECD does have a spreadsheet, so you're able to actually check to make sure that the unique identifier is correct. and matches the correct format for them. Thanks.
Thanks, Austin. All right, so let's go to the next question, and this is how can you upload information about risk assessment in Bolivia? And I think this speaks to exactly what we were just discussing in submit. So we can, here it's asking us if we want to save because we started creating a new record, and we'll say exit without saving. So we see here if the individual that submitted that is a focal point for Bolivia, they would go ahead and just add a new record beside this common format for risk assessments. So that would be exactly how they would create a new risk assessment that was generated by a regulatory process. And then the final question we have in this section here, I will pull up, is on how to update information for laboratory testing and food safety assessment. So that's a great question. These first few ones were about how to create new content and questions about that. But this last one is asking how we update information. And so there's a couple ways that you can go about that, and it really depends on what type of information you want to update. If you are a national user and you want to update a national record, let me pull it up. You can actually go in and click on the title of any of these record types, and it'll take you to a landing page where you can see and manage all of those records. So as a Secretariat user, you see that I have access to many of or all of the reference record content. So if you're a national user, you would see lots of information related to drafts you might have for that record type, what's been published, and any potential requests for action you have. So let's go ahead here. I can click on laboratories for detection and identification of LMOs. And here we see a full list of all of the lab records. If I wanted to edit one of them, I simply would just click on this button over here that allows me to edit that laboratory record. So if you're a national user, again, you can go to the landing page for a specific format of interest and edit records as you wish. But I will say for reference records, because these are managed by the Secretariat, anyone can still go in and make any necessary changes that they want to. So you can even through search, let's say you are looking at laboratory records and search, and let's say you see information that either is outdated or needs to be changed, and you know what that change should be. So you have a couple options. As Austin mentioned during his presentation, we do have a report button at the bottom where you can flag to us at the Secretariat that there's something missing or problematic in the record. But you can also just go ahead and become a contributor to the record and just click edit. So if you do so, and just as a regular registered user on the BCH, you can take part in the submission process for the specific record and go ahead inside and make any change that needs to be made. So I think that answers that question. Austin, was there anything you wanted to add to that?
Just to highlight that after you submit the record, it will go to the Secretariat for validation. So I believe there's a little box that comes up that they can enter some information into. So it may be good just to type maybe a reason, a brief reason. So the Secretariat will review it and validate it. So also if you are a manager of a particular laboratory and you might be alarmed that the people could edit the record, it's not published automatically. It will come to us first before it's published.
Absolutely. Thanks for clarifying that, Austin. And we go through some of those details in the webinar on publishing, but Austin brings up a very good point about workflows in the BCH. So for national records, a national focal point can assign as many authorized users as they wish. So a national focal point can go in and create any record and publish it immediately. But their authorized users can go in and submit any record, and then that would need to be still reviewed, verified, and published by the national focal point. And similarly, as Austin was just pointing out, for all reference records in the BCH, it has a similar process where anyone can submit, anyone can edit. But the workflow process is such that any change or thing that you submit for publication goes to us. We then verify, make any necessary changes, and then publish that information. All right, so I think that wraps up the first block of questions about that are regarding the process for publishing. Let's look at some of the questions that are related to searching. So the very first question is quite general and is, I think, fully discussed in Austin's presentation. What are the different ways to find scientific information? I think just in brief review, the key places to go are directly in our main search tool that we have here, where you can really not just look at all the records that are available, you can really use the filters to find exactly what you're looking for. And as Austin showed in some of his slides later in his presentation, another place that's a very useful place to get a tabular list of all of the living modified organisms, or genetic elements is in their respective registries. And as Austin showed, these are simple tables that show everything that's been published in the BCH. But to really query the information, and if you're looking for something in particular, it's best to use the core search tool for that. So that was the first question, just almost in summary of the presentation, really. The next question we see here is how to share scientific information. And so that's a great question and something we did cover in a webinar earlier this year in January, where we were describing some of our new features on the BCH. But let's say you want to share information on, let's say we're looking at LMOs, and let's say we want to share with a colleague a specific, let's say LMOs, let's see here. Well, let's just say LMOs, genetic elements in organisms, and we want to share all of these records with a particular colleague or user. We can go ahead and use this share button here. And in doing so, we can generate a link and copy and share it with anyone as we wish, or we can also send it by email. So we can just go ahead and input any specific email set of individuals we would like to share it with. Now, let's say we are a national user and we want to post or embed some of this information into a national website. We can use that share button as well. So let's say we have a page on our national site where we want to input, let's say we are Belgium, and we want to input our competent national authorities on a particular page. We can use this share tool to do that as well. So here we can click this embed button. We see that Belgium has 6 competent national authority records on the BCH. And let's say we just put in the domain, generate the code, and then we can copy this and paste it into the content section of that particular page. So this share tool here, which we rolled out last August, allows you to generate links of particular search results or particular records. It allows you to email them, and it also allows you to embed it. Now, let's also say another thing that I think is worth pointing out while we're looking at this, and Austin pointed out in this presentation, but let's say you are interested to keep up to date on some of the scientific information that we have in the BCH. And let's say you're mostly interested in living modified organisms, genetic elements, organisms in laboratories. You can go ahead and save this search. Let's say scientific information in the BCH. And you can select to receive an email when there are new records or updates to records published in the BCH. Now you've selected that. If you go to My Saved Searches, we see now I have a new alert that's set for these record types. And I have my update frequency set to daily. You can decide you want to receive a weekly email that will just give you a summary of all of the new records that are published that meet that criteria. All right, so let's go back to the question slide.
Zachary, can I just
Yes, please.
interject 1 quick second? Also, if you are a scientist or if you publish in the field of biosafety and you would like to share information with us and share it on the BCH, we encourage you to do so. So you could use, for example, the virtual library resource records. So just also to say that we also accept publications and information from users as well, and we would strongly encourage it.
Great, thanks, Austin. So let's see. The next question here is asking, is all information of biosafety and biotechnology uploaded? So that's a good question, and I think it goes to some of the earlier slides in the presentation on the context of the BCH. The information on the BCH is specific to the mandate of the Cartagena Protocol. So the information that's published is really grounded in the needs of the protocol. So it's not comprehensive in the sense that everything's included. It's really specific to what is mandated by the Cartagena Protocol. But Austin, was there anything you wanted to add to that question too?
Just that the Secretariat works hard to ensure that the database is as comprehensive as possible. However, there may be cases where information could be missing. And that's also why we try to link with some of these other databases, and we are collaborating with them to try to ensure that the BCH is as complete as possible within the scope of the Cartagena Protocol.
Thanks, Austin. All right, so the last question here is asking whether there's a place to find scientific information in the form of listed references separately for each GMO, including stacked GMO events. So Austin, I'll pass the floor to you on that one, but I think it's something that we have discussed in terms of how to search for information and how to use the registries.
So in terms of the references for a particular LMO, they can be found at the bottom of the record. So there's an additional information section where I think in my presentation I showed that there were a few links. So here's where the Secretariat works hard to ensure that there are the particular references, so PDFs, DOI links, links to other databases so that you can find references to that particular LMO. But there's also either searching through the registries or searching at the bottom of the page, you can see the stacked events for the particular LMO. So from there, you're able to sort of click through and find the particular reference information that you're interested in.
Great, thanks, Austin. So I see we do have a question in the Q&A asking about how IPLCs can upload information. And I think that the key thing to highlight for that question is that IPLCs or anyone can upload reference records. So if there's any specific information that you would like to upload, you can do so through the common formats that are reference records here. And I see we also have some questions in the chat, but I believe most of my colleagues have fielded those. I see one about whether we give individual countries training on the use of the BCH and benefits for new users. UNEP Jeff does have a initiative through the BCH3 project in which national workshops and regional workshops have been hosted for BCH3 countries. So that initiative exists, and there's a host of material that's available even to countries that are not BCH3. So on the BCH page, you can actually access much of the work that's been conducted by regional advisors and the BCH3 project by just accessing the training materials drop-down menu here. And this takes you to that virtual learning environment where you can access some additional resources that are related to that. I see my colleague here has shared the link to the virtual learning environment in the chat. But through the BCH itself, we do have our knowledge base, which you can access through the drop down in help, but also in the about. And this walks you through this contextual information about the BCH and its context. There is some specific walkthrough information on how to submit information, how to use particular functions and new features. So this is really our big compendium of help material that's directly within the knowledge base. And then if you're searching the BCH and you're not sure how to perform a function that you're trying to do, you can click this little question mark at the top right here. And this is our contextual help pane. And it actually pulls in specific articles from the knowledge base to the page that you're currently navigating. So if you are in the submission form for a record, when you click contextual help, you'll find specific articles that will help you through the submission process. We do also have these tours here. So on some of the key pages on the BCH, you'll see this little icon. So in this case, it's a tour for this dashboard. And this will, in just a few slides, walk you through what Austin and I discussed earlier, essentially how to understand and make sense of the information that you're seeing here. So these tours are a really good way to get acquainted with a page that you don't feel very familiar with. And I think there was also a question earlier about accessing this webinar and past webinars. We do have a specific article in our knowledge base for that. So you'll see a few previous webinars, and this one will also be posted there in the next few weeks. So maybe I'll also request a colleague to send a link directly to that webinar article for users to access. All right, so we are coming right up to the hour, and I see we have also gone through all of the questions we've received. So I think maybe I'll see, Austin, was there anything you wanted to add before we close?
Just to thank everyone for their time and their attention during this hour.
Great. Well, thank you very much, everyone. Oh, I see one last question that came in, and it is asking about the template for the fifth national report. So the fifth national report was approved at MOP. So it is available in the annex to the monitoring reporting document. And it will, we're currently working on the online format. So that's one thing, one of the priorities on the BCH right now is creating that online form for users. So we'll certainly be keeping you updated on that. All right, well, thank you so much, everyone, for joining. And we really appreciate you taking the time. So we'll hope to hear from you soon. And just as I part, I will say we're always available to take questions. So please jot down this email address that you see, bch@cbd.int. If you ever have a particular issue that you want support with, we're here to help. All right, well, thank you so much, and we'll hope to see you soon.